Okay, good morning, everyone. My name is Russ Jasper. Oh, can you hear me? I'm probably loud enough anyways. I'm a PhD student, okay, I'll start, at the University of Bern in Switzerland. And today I will be telling you about some of my preliminary results where I'm looking at genotypic local adaptation in the gut microbes from deer. Just so we're all starting off on the same page, this is how I would envision local adaptation. So it's when you have environmental heterogeneity, and then you have different genotypes or different populations, where the local populations have higher fitness than the foreign populations. And so we have a lot of common examples in macroorganisms. So lodgepole pine local adaptation to climate, or three-spine stickleback local adaptation to salinity. And we know a lot, or we're beginning to know a lot about local adaptation in macroorganisms, but we know a lot less about the process in microorganisms, and especially so microorganisms in their natural habitats. And it's not immediately obvious just because we're beginning to understand local adaptation in macroorganisms that we can just translate that understanding to microorganisms. So microorganisms are different in a lot of ways. For one, their population sizes can be huge, so sometimes on the order of billions. They can have really high gene densities. So E. coli, for example, has a gene density 1 to 2 orders of magnitude higher than ours. Micropopulations can have extreme dispersal rates. So there's this notion that microbes are everywhere. They've colonized every single niche. And then they can be clonal or have really high rates of clonality. And these first 2 points, so extreme population sizes and very high gene densities, these are going to increase a microbial population's response to selection. So maybe that's going to predispose them to local adaptation. So maybe that should be our expectation. But on the other hand, extreme dispersal and high clonality are going to homogenize populations and inhibit local adaptation. So maybe we should not expect to see local adaptation. So the first question for me is, what is our expectation to begin with? Do we even find within species genotypic local adaptation in microbial populations in nature? My second question is, if we do find genotypic local adaptation, what kind of migration selection parameter space are we looking at? Do we only find local adaptation in really isolated populations? Or can we find local adaptation in the face of high gene flow? And then my third question is for the molecular biologists, and that's what kind of gene functions are important for local adaptation in these populations? This is my study system. So we've sampled 10 different gut compartments along the GI tract in deer, and then we have multiple deer. So my ambition is to look at the microbial populations in the different gut compartments and see if I can find genotypic local adaptation to these different environments. And this is de novo assembly metagenomic community, so I have 700 species. So today I'm just going to show you my 2 favorites because I can't fit 700 into 12 minutes. So this was question 1. What's our expectation? Do we even see genotypic local adaptation in these populations? Okay, so this is my first example for you. So it's Quinella. This is my microbe species. And I'm looking at populations in the reticulum. So that's the second stomach. So deer, they have 4 stomachs. So this is Quinella in the second stomach versus the Quinella population in the duodenum. And I'm showing you a Manhattan plot. So it's the position on the X. And then the Y is it's basically our evidence for selection. So it's a log transformed p-value from a contingency analysis at each SNP. So basically, if that's gobbledygook, what I'm showing you is just really high Y values we're more confident that these SNPs are involved in local adaptation. I'm going to highlight this one gene, so HSP70, and I've highlighted all the SNPs in the gene. And this I'm really excited about because this gene has a whole bunch of extreme SNPs. So these are the SNPs that have really extreme allele frequency changes as you move from the reticulum to the duodenum. And then those extreme allele frequency changes are concurrently seen across multiple deer. So this, to me, it looks like a strong signature of genotypic local adaptation in this Quinella species in the second stomach of the deer versus the duodenum of the deer. Okay, I'll show you one other example in Quinella. So it's the same microbial species, but now we're looking at the fourth stomach versus the duodenum jejunum. And I've highlighted 3 genes here because they're really peaky. They look like strong outliers. And then since this is de novo assembly, what I've done is just looked at the orthologs of these genes that have known functions. Then I've stuck up the known functions here. So the 2 red ones are genes involved in replication, recombination, repair, and the blue is involved in cell division and chromosome partitioning. So my very speculative hypothesis right now is that when you look at this Quinella in the fourth stomach versus the small intestines, it looks like there's divergent selection on some kind of replication or cell division. So they're replicating differently in the stomach versus the small intestines. Okay, that's Quinella. That's my favorite species. I'll show you my second favorite. So here's Murray-Baculacea. Now we're looking at the back of the gut. So we're looking jejunum versus colon. And you can see there's a whole bunch of peaky genes again. So a whole bunch of genes that look like they have evidence for local adaptation. I've highlighted 5 here because they're all really peaky and they're all involved in the same kind of processes. So they're all involved in replication, recombination, repair, 2 integrases, 2 excisionases, and a type 3 restriction enzyme. So I'm not a molecular biologist. I don't know what's happening like on the ground. All I know is these genes are really peaky. They're involved in the same functions. So maybe they're being divergently selected for some kind of replication in the jejunum versus the colon. So my first question, what's our expectation? Do we find genotypic local adaptation? And I think yes, we do. My second question was, okay, so we found genotypic local adaptation in some of these populations. What kind of migration selection parameter space are we looking at? Do we only find local adaptation in isolated populations, or can we find it in the face of high gene flow? So here I'm looking at genome-wide FST between populations in the stomach of Quinella. So this was the first species I showed you. So genome-wide FST from Quinella in the stomach versus the Quinella populations in the intestines. And first, if you look at the far one, between deer, so I'm looking at FST in Quinella in the stomachs in one deer and Quinella in the intestines in a different deer. So this gives us the kind of baseline genome-wide FST in isolated populations. And then in the within deer, I'm looking at genome-wide FST from the Quinella in the stomach versus the Quinella in the intestines of the same deer. So they're physically connected, but is there gene flow? So the first thing you can see is that the genome-wide FST, if we're looking at Quinella in different deer or Quinella in the same deer, the FST is the same. So it looks like the Quinella in the stomach versus the Quinella in the intestines, even if they're in the same deer, they're basically isolated. It doesn't look like there's a lot of gene flow. So the signatures of local adaptation I showed you first in Quinella, it looks like these populations are isolated. Now I'll show you the second species, Murray-Baculacea, and immediately you can see the FST is different if it's within the same deer, right? So between deer gives us kind of a baseline for isolated populations. Within deer, it gives us kind of an idea of is there gene flow. And because the FST is so much lower within deer, it suggests that there's a lot of gene flow in Murray-Baculacea from the jejunum to the colon. So the second example species I showed you of those 5 peaky genes, that looks like it's signatures of local adaptation, even in the face of high gene flow. So that was my second question. What's the migration and selection parameter space? And we can find signatures of local adaptation even in the face of high gene flow. My third question was what kind of gene functions? And now I'm going to show you the entire communities rather than just focal species. So here I'm looking at every species in the esophagus that is also in the duodenum. And then I'm taking all of the genes there and looking at all the different functional categories. There's about 26 functional categories. And so on the right-hand side, that's all the genes in the entire community. On the left, it's looking at all the genes that are outliers for local adaptation and looking at their functional categories. And I've just highlighted two functional categories, so defense mechanisms and replication, recombination, and repair, because these are the two functional categories that were significantly enriched for local adaptation outliers. So when we look at the communities in the esophagus versus the duodenum, it looks like they're using genes involved in defense and genes involved in replication, recombination, and repair differently between these two different environments. And then now I'm just showing you the same thing, but just another example. So this is jejunum versus rectum. And it's, again, the entire community. And the right hand is all the genes, the functional categories of all the genes. And then the left is the outlier genes. And now there's 4 categories that are significantly enriched. Okay, energy production, replication, recombination, repair, intercellular trafficking, and translation. So it looks like these are gene functions that are really important for local adaptation in the jejunum versus the rectum. So my third question was, what kind of gene functions are important? It looks like for sure replication, recombination, and repair is involved in a lot of local adaptation in these different communities. So that's all. I just want to thank my lab back home, all of our collaborators, our funding bodies, and you in the audience. Thank you.
That was a fantastic talk.
Thanks so much.
And whoops, maybe it's not on.
Yeah, it's on.
It is,
yeah.
okay. So I guess the whole summary, I like the whole potential convergent evolution on this replication type of thing. It's a difficult question, but what do you think is a selective agent? Do you think it's like the nutrient availability across the GI tract versus microbe?
Yeah, I can speculate, but it's not my field. But so a lot of stuff I showed you was kind of like the very front of the gut versus the abomasum, or very front versus the duodenum, or the other way, like abomasum versus the back of the gut or something like that. Abomasum and the duodenum are super like harsh environments for these guys. Or abomasum, so the fourth stomach. The first 3 stomachs are gentle and not as harsh, but fourth stomach's really harsh. It's really low pH. You have the gastric secretions. The duodenum, you have the bile salts and pancreatic secretions and all that, blah, blah, blah. So my speculative hypothesis would be, if you're living in a really harsh environment, maybe you need to replicate way faster because there's so much die-off. If you look at ruminant richness, it's not the same as monogastrids. It's the species richness and the abundance are really high, then it disappears, and then it goes really high. So my just speculating is maybe it's important to replicate fast in the middle of the GI tract, and if you're at one of the other ends where it's comfy, maybe it's not as important. That's my guess, yeah. But this is preliminary right now, yeah. Yeah?
Are all the species of bacteria you look at acquire in the environment with the feeding, or are they transmitted vertically by the...
Oh, I don't know right now. These are wild deer. And so we had metadata on them, but that got jumbled up. So we don't really know anything. We kind of know metadata, but it's not connected to the deer anymore. I don't know anything about how exactly the communities are structured. All I would say is my expectation, all of the deer we have are sampled from the same day and the same time. So I would have thought they'd all be structured and have the exact same communities, but they don't. So there's a lot I'm still learning. Okay, thanks very much.
Hi everyone, good morning. I'm Charlotte. I'm working as a PhD student in the KU Leuven. So today I will talk about how the gut microbiome of damselflies can actually shape latitudinal differences in their host immunity and in their pathogen tolerance, and how it also can buffer against immunodepression under suppression under warming. So first, many species show actually a big trait variation along environmental gradients. And because these traits tend to shift actually along these gradients, a framework has been developed, which is called the pace of life. And it actually says that species or populations with a slow pace of life tend to develop and grow slowly, but have a lot of energy left to put into immune functioning and pathogen tolerance, whereas fast-paced populations or species are more fast-growing, faster developing, and therefore have less energy left to put into immune functioning and pathogen tolerance. In our lab, we tend to work a lot with the damselfly Ischnura elegans, because this damselfly has a big differentiation in pace of life alongside a latitudinal gradient. So, for example, populations in southern Sweden, which we call the high latitude populations, are slow-paced. They grow and develop slowly because they actually have a shorter growing season due to the lower temperature, so they tend to grow slow and only complete one full generation every 2 years. Whereas populations in the southern France, which are called low-latitude populations, they are fast-paced. They grow and develop very fast because they have a longer growing season. Due to also a higher temperature, they can grow fast and actually have or complete multiple generations, like 2 or 3 generations every year. We now are currently also looking into the gut microbiome of these larvae, because multiple studies have been showing that the gut microbiome can actually also affect life history, physiological and behavioral traits, and these make up the pace of life of these organisms. So potentially the gut microbiome can actually impact the pace of life of these organisms. And therefore, we can predict that it might also affect their immune functioning and even their pathogen tolerance. Also, what we can do with this setup is we can actually predict or try to predict how these high-latitude populations will respond to warming. And for this, we use a space-for-time substitution approach. When these high and low-latitude larvae or populations are actually adapted to their own local thermal regime, so to their own temperature, which is around 20 degrees with 5 degrees temperature fluctuation during the day in the water in the high latitude, and 24 degrees and 10 degrees fluctuation in the southern France, in the low latitude region. So when they are thermally adapted to these temperatures, and since it is predicted that under global warming, actually the current thermal regime of 24 degrees will actually be the future thermal regime at the high latitude, we can try to predict the response of these high latitude larvae, how they would adapt to warming, if they're obviously also able to adapt to it, by just looking at the current phenotype of the low latitude larvae, because they are actually already adapted to the temperature, because they have already experienced this warmer temperature for a long time. So to make it a bit more clear, because it's a little bit difficult to explain, so if we would expose both the high and the low latitude larvae at the 2 temperatures, we can make some comparisons. When we compare the high latitude larvae at their own current colder temperature with the high latitude larvae put at a warmer temperature, we can look at the thermal plasticity of the host. But when we compare it with the low latitude larvae at a higher temperature, we can actually look at both the plasticity and also the evolution of the host. We can make predictions about that. And now, because we're also looking at the gut microbiome, we wanted to actually insert this into the space-for-time substitution approach. So we now not only have the high and the low latitude host and the cold and the warm thermal regime, but we also have the high and the low latitude gut microbiome. So if you then do a transplant with the microbiome, we can actually make these comparisons. When we compare the high latitude larvae with their own cold adapted microbiome at the colder, their current thermal regime, with the first arrow going to the high latitude larvae, but with actually a warm adapted microbiome at the warmer thermal regime, we can look at only the thermal plasticity and evolution of only the gut microbiome. When we compare it with the second arrow going to the low latitude larvae, but with a cold adapted microbiome at this warmer temperature, we can look at the thermal plasticity and evolution of only the host. And then when we look at the last arrow compare it with both a warm-adapted host, a warm-adapted gut microbiome at, of course, the warmer thermal regime, we can look at the thermal plasticity and evolution of both host and microbiome. So with all this information, we set up some objectives, and we wanted to actually check whether the gut microbiome would causally and differentially impact the immune functioning and pathogen tolerance of these damselfly larvae, because they differ a lot in pace of life, and whether the gut microbiome and also the host would contribute to the thermal adaptation and evolution towards future warming. So for this, we set up an experiment whereby we used these high and low latitude larvae. We transplanted their gut microbiome between them and also at the two temperatures, and afterwards exposed them to a pathogen, a pathogenic strain of E. coli, to actually see what happens with their immune system functioning and also their pathogen tolerance. So these are the combinations that we have. So we have a recipient latitude, which is the high or the low latitude host. Then we have the high or the low latitude donor gut microbiome. Then we have the two thermal regimes, the cold and the warm one. And then we have also the pathogen exposure or control solution or the E. coli solution. So in total, when we have a fully crossed design, we ended up with 16 treatment combinations, and the experiment itself looked like this. So first, F0, it means like the final instar larvae, because it's the last larval stage of these damselflies in the water, and afterwards they will emerge actually into a terrestrial adult. And when they reach this stage, we expose them for 2 days to an antibiotic mixture to get rid of the bacteria inside their gut. And thereafter, did the gut microbiome transplant, whereafter we exposed them for 4 days to an E. coli solution or to a control medium. And after or during these 4 days, we measured their growth rate by checking their mass, and we also checked if they survived or not to have an indication of the mortality. After the experiment, we did some physiological analysis, because we looked at phenol oxidase. It's an important enzyme in the, for the immune functioning of insects. So we have an immune function parameter. And we also looked, dissected the gut for the gut microbial analysis, that we know which bacteria were inside the gut. And we also looked at the E. coli burden. It's actually the E. coli strain that we use. If you put it on a special medium, it's colored pink. So then we could really take out the gut, put it on a medium, and count how many E. coli cells were inside the gut. So the more E. coli cells, the higher the E. coli burden, and the less tolerant that the larvae actually were to the pathogenic strain. So then we can go over to the results. So first, maybe on the x-axis is always the latitude of the recipient. Y-axis response variable. And then on top you can also see the different other treatments, like the temperature, the latitude of the gut microbiome, or the E. coli treatment. So first we saw that there was indeed a difference in pace of life between these high and low latitude populations, because there was a big difference in growth. So the low latitude populations did indeed grow much faster than the high latitude populations. So indeed they were fast-paced. And also the immune functioning was much higher in the high latitude populations. So again, high latitude populations, less energy into growth, but more energy left for immune functioning. Then we saw that they were also thermally adapted to their local thermal regime, because when we see at their growth rate that warming actually reduced the growth in the high latitude populations, but increased the growth in the low latitude populations. This actually just means the high latitude populations are indeed doing better at their colder thermal regime. Low latitude is growing better at their warmer thermal regime. The immune system functioning decreased actually at the high latitude populations under warming. So again, for high latitude populations, warming is stressful. A lot of energy has to go into defense against these warmer temperatures, and less energy was left for immune functioning. Then for this immune functioning, it was pretty cool because we could disentangle actually the contribution of the host and the gut microbiome towards future warming, because we found a three-way interaction between the latitude of the recipient, or the host, and their microbiome and the thermal regime. So here we can make these comparisons. So first, when only the host, or the host and the gut microbiome would thermally adapt or evolve to a higher temperature, we saw that there was a big reduction in immune system functioning. But when only the gut microbiome would evolve, the reduction was less, and this was actually equal to just a plastic response. So this means that when only the microbiome would thermally adapt or evolve to a higher temperature, this would actually be better for the host than the host itself would also evolve. And then we also looked into the effects of the pathogenic E. coli strain. We see that there is indeed an effect of mortality, a mortal effect, and it was much bigger at a higher temperature. This is because both a higher temperature and the E. coli, it was just too stressful for the larvae, and that's why it required too much energy to defend. And we also saw that the E. coli burden, so the amount of E. coli cells that ended up in the guts of the larvae, was much higher in the low latitude larvae. So again, fast pace of life, high amount of energy towards growth, less into immune functioning, and they ended up with more E. coli cells in their guts. Okay, cool. Then we can also then we found actually that the high latitude gut microbiome was more positive for the larvae. So, and this is visible in this figure. If we have the low latitude larvae and we just give them a high latitude gut microbiome, so a slower gut microbiome, their immune functioning increased, and this caused that their E. coli burden dropped a lot. So just by having the high latitude gut microbiome, this was much more beneficial for them in terms of their immune functioning. So to conclude, the gut microbiome does causally contribute to the immune functioning and pathogen tolerance in these larvae. Thermal adaptation and evolution of only the gut microbiome might be more beneficial in terms of their immune functioning under warming. And to understand evolution of populations under warming, we really not only have to look at the host, but also have to dig into this gut microbiome because it can do a lot of things. So that's it. Oh, wow. Yeah, so there is the overlap after a transplant is around 50%, because of course, they can't take up everything. But there is a nice difference in, if you look at the beta diversity, you really see that they differentiate in the high and low latitude clusters. And with temperature, you also see a difference. Yeah, of course, it was too much to add here, but there is some interesting stuff happening. For example, when you put the high latitude normally at cold temperature, you put them at a higher temperature, then you really see a shift of the gut microbiome towards that of the French ones, of the low latitude. So yeah, also here for the results, you see like the switch for a higher immune system functioning and stuff, and we did see some bacterial species change in density, and potentially these can be linked to a higher or a better nutritional functioning in the larvae and stuff like that, but we still have to look a bit deeper into that. Okay. Good. Oh, sorry.
I may have missed this in the intro, but how do they acquire the microbiome? If everything is coming from the environment, is your model, like say, the global warming, high latitude population can assemble something from the environment, and how does adaptive plasticity?
For this experiment, we really did it in the lab. So we just sampled from high and low latitudes, but then eventually we did a microbiome transplant really in the lab environment. So just in clear water, we took guts from the other latitude and then put it in the water for the larvae. Yeah. Maybe later, yeah.
Thanks.
Thank you so much.
Okay, my name is Donald Price. I'm going to give a talk today about some microbiome research in Hawaiian picturing Drosophila. An important colleague of mine and Joanne Yu at the University of Hawaii, who I've been collaborating with, and her postdoc Matt Medeiros, and two of my students, Christian and Michelle, have been instrumental for this research. And I'm going to quickly give you a little bit of highlight of two previous studies that have been inspirational to me. Joanne's work on Drosophila melanogaster using wild flies in germ-free melanogaster. That's sort of in the melanogaster world, the way one does microbiome research. And then another study that Joanne led, but it was mostly done in my lab on Drosophila grimshawi picture wing, where I call it a microbiome removal using antibiotics and antifungals. And then the main study will be Drosophila baeaceti, where we did a replacement microbiome from two populations of Drosophila baeaceti. So these studies are founded in Hawaii. Joanne's work is on North Oahu. Grimshawi comes from Maui, and baeaceti comes from the Big Island. And so Joanne's work in Drosophila melanogaster, using germ-free melanogaster, she went out, had her postdoc go out into the field and collect wild microbiome from the feces of flies at two elevations on Oahu. So W1 is a low elevation and W2 is a high elevation. You can see about 300 meters apart in elevation. And she sampled the microbiome of the feces of flies from here, and they differed, and they did it over the period of the experiment in the lab, sort of early, right when they got the flies from the wild, and then later near the end of their study. And you can see that she's looking at both the bacteria and the fungi in here. The fungi were really different in their diversity between the two locations. The bacteria is somewhat divergent between the two locations. And the locations differ in rainfall, elevation, and temperature a little bit. And so the main experimental design is to get those feces from two populations of flies, seed the food, allow them basically to poop on the food, and then use these germ-free melanogaster that are all from the same population of melanogaster, split them, of course, into these two environments with the different microbes, and then grow them there for 10 generations, and then measure phenotypes. And I'm just going to highlight a couple of the assays that she did. But basically, the wild microbes alter the reproduction and survivorship of these germ-free melanogaster that start out that way. And so the high elevation is W2, right? So the W2 microbes increase the reproduction of females. They lay more eggs and they lay them faster. And they're more fertile. Essentially, more offspring are produced when the same types of melanogaster are given microbes from W2 versus W1 population. And then just the opposite for survivorship. And here she did a desiccation stress experiment. So when they're given W1 microbes, the females and males, and I'm just showing you the females here, survive longer in a desiccation stress tolerance. And then they also have more fatty acids that might allow them to do that survivorship better. So that was the main result, a life history kind of study. Wild microbes alter the reproduction and survivorship of phenotypes, fertility, fecundity, survivorship, fatty acids, weights, just about every trait that she decided to measure. And then there is kind of life history trade-offs. So microbes from one location increase reproduction, but decrease survivorship under especially stressful situations. And then the second study, this was done largely in my lab, although Matt was doing it as a postdoc with Joanne. And this, I call it the microbiome removal study. And what they decided to do was use some antibacterial and antifungal to subsequently remove the bacteria and the fungi from Drosophila grimshawi and see what happens to them. Same kind of design. What we found, or what they found, was the antibiotics, or the removing of the bacteria, really had no effect on fecundity for the females when they're given to the females, or when they're given to the males, a little bit when given to both. But the fungi, again, really reduce female reproduction more so. And then when you give both antibiotics and antifungals, they really reduce their egg production here. And then same thing with mating. So when you do this, the antibiotics, removing the bacteria, really had no effect on mating compared to the controls. But again, the antifungals, removing the fungi, really reduce female reproduction. And then when you give both antibacterial and antifungal, they reduce both matings when given to males only or females only, but particularly, again, for females. So the summary of this was antifungal treatments really reduce fecundity and copulation, especially when given to females. fatty acids were greater in antifungal treated females and males. So that's what inspired this research. I said, well, I want to go more wild. So we went out and got Drosophila baecidi from 2 locations, about 300 meters apart in elevation, a high elevation and a low elevation. They differ in about 300 meters elevation, or a little bit higher, in a wet rainforest where it's fairly high humidity and cooler, a little bit difference in temperature in these locations. And this is where we go on the Big Island to collect them. And that's how we collect them, little flies on a sponge, and we capture them. And so the design was collect these 2 populations from these 2 locations, collect the microbiome or the microbes by feces collection from these same wild flies, bring them back to the lab, raise them at a common temperature of 18 degrees Celsius, 60-70% humidity. And here's some of the microbiome results from wild flies. The bacteria differ, particularly in species abundance. The alpha diversity, so alpha diversity is really basically the number of taxa that you'll find there. And there's a little bit of difference in the beta diversity, which is kind of the relative frequency of different taxa in the environment. The fungi didn't really differ in the alpha diversity, but differed more so in the beta diversity, or the relative frequencies of the different bacteria taxa. And this you can look, if you want, the rank order of taxa. I rank ordered them for the most common in Ola'a for the top 10 taxa. You can see the bacteria a little bit different. There's some bacteria more common in Ola'a versus is Tom's Trail. Fungi are more different. There's some fungi that, at least in our samples, weren't found in one location, but were fairly high in frequency in the other location. So then we come in and did the experimental design, kind of similar to the damselfly study, a couple of factor design, the microbes from 2 different locations, the low and the high elevation, and a temperature treatment, temperature acclimation treatment of 18 degrees Celsius, which is the controlled temperature, and 24 that we think would be a stressful environment, and then did all the particular interactions. And these flies take about 3 weeks to be sexually mature. And so a lot of different traits differed. So survivorship, just temperature, it appears to be stressful to them, a trend for significance in both populations within there. But then when we gave them the microbes from the different locations, if they get microbes from Tom's, which is a lower elevation site, they survive better at both 18 and 24 degrees Celsius in this sort of acclimation temperature that they were in for several weeks. And then egg production. Here, temperature is stressful. They lay about twice as many, or have twice as many eggs at their controlled temperature versus the 24 degrees Celsius. So females are feeling stressful, or at least not producing eggs in the 2 places. And then now just the opposite, sort of like Joanne's work, when they're given a lot of microbes, they produce more eggs compared to when they're giving the microbes from Tom's Trail, and that occurs again from both populations. Even though the populations produce different amounts of eggs, when the microbes are given to them at both, again, 18 and 24 degrees, Tom's produces more eggs. And here we did now a temperature stress experiment. At the beginning of the temperature stress experiment, we take 10 minutes and look at their movement using this program called EthoVision that can analyze movement very quickly in lots of flies at the same time. Here they move about twice as much, even though they don't move a lot. Picture wings don't move a lot. Twice as much when they're given microbes from their home location versus the other location. So you can see Ola'a with Ola'a moving more, Ola'a with Tom's microbes moving less, and vice versa for Tom's flies. And then when we do the CT max, which is a physiological measure for when they're going to die, as we ramp the temperature up from this 18 degrees Celsius up to past 35, 37 degrees Celsius, very slowly, again, using this EthoVision program. It was an interesting reaction. The flies from the 2 populations responded differently to both temperature and microbes. I don't fully understand this one, but Tom's flies were very sensitive to the microbes. A lot of flies seem not to care. I don't fully believe it, but that's what the results show us within here. So what we find from the baecidi population is survival and egg production during acclimation was greater at 18 versus 24, so at a stressful temperature. Survival was higher with microbes from toms. Eggs production greater for microbes with Ola'a, so a type of life history trade-off going on here, reflecting back to Joanne's melanogaster work. General activity was greater in flies given microbes from their own location. And then the CT max was kind of a complex interaction of fly location by temperature acclimation. And so the general results of these three studies is yeah, microbes matter. They affect every trait. This is a new field for me, so I'm a convert to microbiome research because of that 1 minute. Thank you. Wild microbes showed survival and reproduction trade-offs, Joanne's work, and then our work with Bay-CD. Microbes that increase reproduction seem to decrease survivorship and vice versa. The fungal component, which I'm learning from Joanne and others, is kind of a newer area. Don't forget about fungi, at least in melanogaster and Drosophila. They're especially important for female reproduction, it appears. And then the motivation behind a lot of this study is conservation. Hawaiian Drosophila biodiversity is deteriorating. This is why I got into temperature tolerance and microbiome. There's 12 species that are endangered species. And so we're now looking to implement sort of microbiome protocols and temperature acclimation protocols to see if that helps in reintroduction and relocation of species. A very common thing in insects is they fail. Reintroductions typically do not hold, and we don't know why. We're thinking now it might be microbiome might matter. Thank you.
So it seems like the fungi are affecting health and behavior. Do you have any insight into what it's doing metabolically for the flies?
Yeah, I'm personally surprised by it. We know, of course, Drosophila are fungi eaters primarily, so that doesn't surprise me they don't get their energy from there, but why they're affecting something like egg production. So when the antifungals are given to them, the females pretty much don't produce any eggs. So it's probably, and they did some other studies I didn't talk about to try to see if it was nutritional. It doesn't appear to be nutritional only. There must be signaling molecules interacting with hormones, starting to look at some of the hormones that are probably important. Yeah, mechanisms is the next place.
Any clues on egg viability versus egg number?
Yeah, Joanne looked at that in the melanogaster study in the Grimshaw. I've not done that in Bay-CD, but yeah, it would be good. Those are harder studies. Yeah.
I know Drosophila kind of is impacted a lot by Wolbachia. Actually.
Yeah, these both, the Bay-CD and Wolbachia.
So have you, in the future, you planning to look at all about maybe?
That would be cool, right? Yeah, we've done some work with the Wolbachia. Probably when we give them the antibacteria, they're probably removing the Wolbachia. We've got metadata out, metabarcoding data to come in to see what actually is in the flies once they've gone through this treatment. I don't have that data yet.
Do you have a sense of if the antibiotics and antifungals have any like direct effect on the fly?
Yeah, really good question. So in the Grimshaw study, they actually live longer. So these were actually chosen very carefully. They went through a couple of different ones. That's why the captain was chosen as an antifungal. They actually increased survivorship of the flies, so we don't think they're toxic. But yeah, it's a good question. I want to try to get away from it when flies emerge. They don't have any bacteria, but they have lots of fungi. So yeah, you probably still have to give them antifungal.
Thank you.
All right, so hey everybody, I'm Cam Hudson. I'm here from the Swiss Federal Institute for Aquatic Science and Technology, and I'm going to talk to you about some of my postdoc research with Christoph Warburger, who is right there, if you want to meet him later. And so we've been studying the relationship between temperature and coevolution between hosts and parasitoids and aphids and their endosymbionts. And so many of you are probably aware that aphids and many other insects carry bacterial endosymbionts. Some common examples of this would be Wolbachia or Spiroplasma. And these endosymbionts can either be horizontally or vertically transmitted. So they can be passed from parents to offspring, or they can be acquired from the environment or transmitted between individuals through sex, for example. And when we talk about endosymbionts, we refer to them as either obligate or facultative. So obligate endosymbionts are ones that have become essential to the life history of the organism. In the case of aphids, they have one which helps them synthesize amino acids, and if they don't have this, they'll die. Whereas facultative endosymbionts are not present in all members of the population, and they can provide various costs or benefits. The cool part about the benefits is that sometimes they can do things like aiding with adaptation to thermal stresses, so heat stress, or protection from pathogens, fungi, or parasitoids, for example, in this case, and also for host plant defenses. And so one thing that interests me is why are some endosymbionts facultative? And so I've kind of put up a series of reasons why I think they might be. And so the first one here is that there could be unreliable transmission from parents to offspring. So if the mother is not always able to pass the bacteria along, you'll see the frequency change in the population over time. And then down in the bottom right, or bottom left there, the environmental context can matter. So under circumstances where, for example, it's hot, if an endosymbiont helps the animal survive, whereas those that are uninfected will die, you'll see an increase over time in the population. Similarly, when it comes to things like parasitism risk in an area where there's lots of parasitoids, if you have a defensive endosymbiont, this will protect you from getting killed by wasps, whereas those who don't carry the endosymbiont will get infected and wasp larvae will kill them. And finally, all this kind of boils down into fitness trade-offs. So there's often costs to carrying endosymbionts, even if they are beneficial. And so an organism which may not have it could produce more offspring than one that does have it, again, but it's all contextual. And so the system we work in is the black bean aphid, which is a widespread agricultural pest through Europe, and it's also introduced to many parts of the world. And their parasitoid, Lysophlebus fabarum there, you can see it stinging an aphid, so we have an adult and some nymphs. And then the third player in this game is the defensive endosymbiont Hamiltonella defensa, which you can see in that picture there. It's highlighted in red. And so this is in the hemolymph of aphids. And the way the system sort of works is if you look at the blue cycle here, normally a wasp will sting an aphid and lay an egg inside of it. If the aphid does not carry an endosymbiont or this endosymbiont is inactive for some reason, the wasp larva will grow inside the aphid, eventually killing it, turning it to a mummy, and hatching out as a new wasp. When aphids do carry this defensive endosymbiont, which carries a phage that encodes a toxin, which is lethal to wasps, the wasp will die early in development as a larval stage, and then the aphid survives, which then leads to passing on more baby aphids containing the endosymbiont. And what we find interesting here is that the different strains that we see in nature vary in both the cost that they impose on the aphid and the amount of protection that they can provide. So if you look on the left of each graph, we have the uninfected individuals here. And so the lifespan of a normal aphid is somewhere between 25 and 28 days. When they're infected with the blue arrow, oh yes, so the colors represent the different strains that I'm going to be working with, and I'll describe later in the experiments. So when they're infected with haplotype 1, the blue arrow, we see not a huge change in lifespan and actually a pretty good level of protection. So when exposed to wasps, very few individuals are killed compared to the normal of about 40% to 60%. In contrast, when you look at haplotype 2 or 3, the purple or green, these seem to impose a pretty strong cost on lifespan. So for example, in some cases, they can actually lose about 10 days of their life. And these may not actually be that protective. So when exposed to wasps, you see the green arrow there, about 40% of aphids will still be killed. So there's a lot of sort of fitness trade-offs here with the infection rate and the protection they provide. One thing that we can sort of see is there's a pattern with the titer, so how much bacterial density an aphid has in its body and how transmissible the bacteria is if you try to artificially infect aphids. So if you stab an aphid with a needle and then stab another aphid, generally if it's more abundant in the body, you're more able to infect the aphid. So this leads us to think about the transmission also between parents and offspring when they're passing it along. When we look at our lab cultures, or generally in nature as well, the transmission is typically 100% under ideal conditions. So aphids will almost always pass along the bacteria when they're happy and doing normally. But an anecdotal evidence from our lab showed that when we lowered the temperature of our incubators, some of the aphids started to lose their infection, which led us to wonder if maybe temperature plays a bigger role than we think. Some field studies have also shown that over time, throughout the season, the rate of Hamiltonella can increase over time, and this is not always necessarily related to the number of wasps present. So temperature also seems to increase, like hotter temperatures lead to higher levels of infection in populations. So this led us to our first sort of research question, is whether or not maternal transmission of Hamiltonella is temperature dependent. in the black bean aphid. To test this, what I did is I had 2 aphid clones that were collected in Switzerland that were naturally uninfected. And so then we transfected these aphids with the same 3 strains that I mentioned earlier, haplotype 1, 2, and 3, which were found from France, Switzerland, and northern Italy. We then raised these clones under 3 different temperature conditions, so 10 degrees, 20 degrees, and 27. We were hoping to use 30, but then 30 was too hot and everything died. So we went back down to 27. And the way that we would perform this experiment is every generation, I would bottleneck the population down to 3 mothers, and then we would have 3 replicate plants per condition. And so as you can see there, the generation time also changes quite a bit. So when you keep aphids colder, they grow a lot slower, whereas when they're in the hot condition, they can have a full generation in about 4 days. And so jumping to some results, to sort of explain this here, this is a plot showing the survival of the 2 clones and their various treatments. So the yellow lines, as you can see, that run the whole length of the plot, this means that the line survived the entire experiment. So something that went from F0 to F6, it's a full line. If it stops anywhere there, that's where the line died. And so what you might notice right away is that we saw a clone by temperature effect. So clone 1, aphid genotype 1, seemed to be cold sensitive because everybody died before the end of the experiment, whereas clone 2, the uninfected and some of the infected lines did fine in the cold. And then we had sort of the reverse with clone 2 being sensitive to heat, whereas if you look at the left corner, the uninfected version of clone 1 was sensitive to temperature, but when it was infected with Hamiltonella strain 2 or 3, it actually saved it from dying. So they did quite well, which means potentially these strains can provide some sort of thermal benefit. We also had one interesting thing happening at the F3 generation that you can't see here, but as we ran the experiment, we screened the individuals to see whether they were infected or not. So I collected both mothers and sisters from the experiment. And what you can see there in that one plot with a lot of red at the top with haplotype 2 is that in cold conditions around the F3 generation, we had pretty much most aphids being cured by then. So blue is infected and red is cured. And then when we transfer these back to 20 degrees, the infection doesn't seem to come back. So the aphids are actually cured by being cold. And if you repeat this with another strain, we have the same pattern. So what we wanted to ask then was whether this is just because cold reduces the titer of the bacteria within the aphids. So we did some qPCR. And what you can see there on the left is that very quickly the bacterial density of the orange and purple lines, which are the haplotype 2, the one that did go extinct, declined very quickly. So by the 3rd generation or so, they're not passing it anymore, and the infection is pretty much eliminated. Here's when you move those individuals from the F4 generation back to 20, it doesn't recover. So the aphids are cured. Given that there's a large range of temperature variation in nature, this led us to think maybe this would influence the frequency of infection across Europe, where we see wild populations. I'll note that there's also parasitoids throughout the range as well. So it's hopefully they're both present at similar rates. And so what we did is between March and July of 2023, we sampled 20 sites about 200 kilometers apart, starting in Sicily and ending in Umeå, sort of in northern Sweden. And we collected aphids from fava beans, sugar beets, and goosefoot. The plan was to collect about 50 individual aphids, sort of unique clones per site, and then bring back some live colonies for further experiments. And so what you can see here is a pretty strong decline of infection prevalence. So in the south of Italy, we had some populations, like in Naples, where it was about 92% of aphids are infected. Whereas in the north, once you get north of Uppsala, they don't carry Hamiltonella anymore. And you might also notice from the color of the pie charts, when you're below the Alps, we see all 3 strains, the ones that we were using in the experiment, are present, whereas north of the Alps, it only seems to be haplotype 2 that is found. So because I'm obsessive, I collected, I used 19 different unique genotypes from the transect and reran the cold part of the experiment again to see how sort of applicable this result would be. And we only ran it for 3 generations this time because we sort of saw that's when the infection seems to drop off. And if you can see there, the haplotype 1 and 3 fully infected after 3 generations, whereas haplotype 2, we had 7 clones fully cured 5 partially cured, and only 1 that was still infected. But it doesn't seem to follow the same latitudinal patterns. It seems to be specific to this one strain. So I guess what does this mean? Oh, weird. Okay. It says, what does this mean? 1 minute. Perfect. So I think the interesting part about all this is that we have a temperature-mediated decline of resistance to parasitoids. So in the north, where there is no defensive symbiont, the sort of coevolutionary relationship that's going to be interesting is between aphids and parasitoid wasps themselves. So there's going to be selection for the sort of arms race between aphids and wasps. Whereas in the south, because aphids carry this bacteria that helps them, the relationship between wasps and the bacteria and sort of evolving resistance to different strains and strain diversity, begetting more diversity, is worth looking at, I think. So as always, warming climates could lead to some rain shifts, and perhaps we'll see more infections of aphids going forward, which could cause problems for agriculture since these wasps are used to try to control aphid populations. And so just wanted to say thanks to my partners in crime for this sort of large field sampling effort, and I'm happy to take any questions. Yeah.
Thank you for the great talk.
Thanks.
Do you know about co-infection with other symbiotic bacteria in study populations when you saw such a nice line?
Yes. So there's not many. We screened everyone for Rigella, and so Rigella actually peaks around northern Italy, but co-infections are quite rare. So we actually only had, I think, of the 1,000 aphids, there were about 25 co-infected with Rigella, and then here from the rest of the populations, there were we screened these other endosymbionts, and so it doesn't seem to explain the pattern very well. Yeah. Thanks. Yeah.
Can aphids screen their own endosymbionts? Can they find them?
I'm not sure. I don't think so. But that's a good question. Yeah.
In the beginning, you showed the different effects of the 3 haplotypes on the survival.
Yep.
And I wanted to ask if that's a general observation with all kinds of wasps you use, or is that maybe genotype specific? So in terms of what type the wasp, like what species or what genotype the wasp is?
So there is specificity between both species and wasp genotype. So some wasps are completely unaffected by Hamiltonella, and others, even some genotypes of the same species, have resistance to specific toxins that are encoded by the different viral genes. So it is more complicated, but yes. Yeah.
Yeah, great talk.
Thanks.
So have you had a look at any of the genes that might be involved in the different haplotypes that could cause the sort of stability of the family stability of the different hosts?
I have not, but we have a new postdoc who will be doing that once I'm gone. So maybe in a year or two he'll have something to say. Thanks.
Hello, I'm Bob Week. Sorry, just getting set up over here. I'm a postdoctoral researcher working with Brendan Bohannon at the University of Oregon. Brendan is an empirical microbiome biologist, and I am a theoretical evolutionary ecologist, and so it's been really fun to have this feedback between his crazy ideas and me trying to make those ideas more rigorous. And so what we've been working on is this really fun new theoretical framework for understanding how microbiomes can mediate the adaptation of the host organisms they associate with. Let's get into it. So nearly all plants and animals have associated microbiomes, and these microbiomes mediate many important host traits. For example, the human gut microbiome mediates the absorption of nutrients, and also the fish skin microbiome mediates resistance to infection by pathogens. And so as a consequence of this, microbiomes mediate variation of host fitness. And in addition, microbiomes can be transmitted either lineally from parent to offspring, through social contact, or through the environment. And so this combination of microbiome-mediated fitness differences in the transmission of microbiomes across host generations provides the raw ingredients for microbiome-mediated adaptation. And so microbiomes can influence evolutionary processes in general, and this has important applications for medicine, agriculture, and conservation. But in spite of this importance, we know hardly anything about the process of microbiome-mediated adaptation. So the current state of the art of the field suggests that we can partition host trait variation, P, into components due to genetic variation, G, environmental variation, M, and variation of the microbiome associated with the host across hosts, right? But we know hardly anything about the processes that shape the phenotypic variation that's associated with microbiome variation. So this suggests that there's a need for a new theory. Okay, so the goal of this research project is to try to journalize classical quantitative genetic theory to account for phenotypic variation that's explained by variation of the host microbiome. And so we're going to take inspiration from genetic evolution by starting with this additive model. So Z is going to be some trait value of the host organism, and we're going to decompose it into a bunch of additive effects. And so in the classical quantitative genetic realm, the index I would be a locus along the genome, right? And then alpha would be the additive effect of the alleles at that locus. And then this number n would be the count of alleles at that locus. For example, with diploid organisms, n could take values 0, 1, and 2. So for the microbiome, we're just going to sort of reinterpret all these parameters. And so instead of the genetic locus, the index I will be the ith microbial taxa in the microbiome. And then alpha I would be this per capita added effect of the ith microbial taxa. And then Ni would be the abundance of that microbial taxa, or more realistically, the relative abundance of that microbial taxa, which can actually be measured. Okay, so in spite of this like fun mapping between the genome and the microbiome, there are some important differences. So the genome is lineally transmitted from parents to offspring, but the microbiome is not necessarily linearly transmitted. It can be, like as mentioned earlier, it can be transmitted through social interactions, and it can also be transmitted via shedding into the environment and subsequent acquisition from the environment. In addition, the genome is this really high fidelity thing. There's relatively low mutation, so genetic information passed from parent to offspring tends to like have a high resemblance. In contrast, the microbiome are these highly dynamic ecological communities, right? And so there's a lot of variation that's produced throughout the process of the colonization of host organisms, but then also the dynamics of the microbiome that occur throughout the development of the host, right? And so what this is really pointing at here is that there's two main components in thinking about adaptation, right? There's selection, but then there's also inheritance, right? And so what this is pointing at is that the concept of inheritance for microbiome-mediated traits is quite drastically different from just the classical genetically mediated traits. And so for the rest of the talk today, I'm going to be introducing a model of microbiome inheritance. And so this model is going to have three main components. There's going to be transmission, this lineal transmission of microbes from parent to offspring. There'll be microbes being shed into the environment, and then subsequent acquisition of microbes from the environment, which forms the environmental transmission of microbiomes. And for now, just for simplicity, I'm just going to ignore social transmission. Right. And so this general diagram that I'm going to introduce the model with will have time running from top to bottom. The parent host organism is this blue circle on the top left. And then the environment that the hosts hang out in is this brown rectangle. And so the offspring is at the bottom, and the green circle inside the offspring is a microbe, right? And so we're going to do this sort of like microbe by microbe, and we're going to ask where did the ancestors of this microbe come from, right? And so I'm just going to keep it very, very simple and say that with some probability L, this microbe has ancestors that come from that offspring's immediate parent, right? And so with probability 1 minus L, then that microbe must have come from the environment. Okay, so L creates this parameter that forms this measurement of lineal inheritance directly from parent to offspring, right? But the microbe could have also come from the environment, and in this model, there's hosts can shed into the environment, and so the environmental microbiome can also be shaped by host shedding, right? And so we'll look at the brown circle in the bottom here is the environment that the offspring experience. And so now we'll look at a microbe in the environment that the offspring experience, and we'll say that just with some probability epsilon, that microbe has ancestors from the parental environment, and then with some probability 1 minus epsilon, the ancestors of that microbe come from the sort of pooled parental microbiome that like one of these parents shed this microbe into the environment at some point. All right, so this creates our model of microbiome inheritance, right? So we have this sort of bifurcating probabilistic tree. So the offspring, again, is the red circle at the very bottom. So we have different routes at which this microbe can be inherited from the parental population, right? So I'll use this lowercase m to denote the trait value of a microbiome-mediated trait. So m prime is the offspring trait value. M is the trait value of that offspring's immediate parent, its actual biological parent, right? And then this m bar star is the average microbiome-mediated trait value of the parental population. The star is to indicate that we're post-selection, but I'm not really talking about selection in this talk right now. And so we want to calculate what is the microbiome-mediated trait value of the offspring given this model, right? And so there's going to be a component that's due to the lineal inheritance, right, which is just going to be L times the parental value M. And then there's going to be this component due to this environmental acquisition, right? So 1 minus L times 1 minus epsilon times M bar star, right? And then there's all the other stuff where it gets from the environment. That's like the non-heritable component of this microbiome-mediated trait, right? So the heritable components here is not just L, but also this 1 minus L times 1 minus epsilon provides this route of this sort of environmental inheritance. And so if we average across all the offspring and the offspring population, we can then calculate, based off of this model, what the consequential average microbiome-mediated trait value should be. So with this model, we've already arrived at our first really interesting result, which is that under this model, we can have adaptation, we can have an evolutionary response to selection without any parent-offspring transmission. So we don't need to have any parent-offspring resemblance at all for there to be a response to selection. And so we find this by setting our lineal transmission or lineal inheritance parameter equal to 0, right? And so all the green stuff goes away. And then the 1 minus L just becomes 1, right? And so we still have this response to the selected microbiome-mediated trait value, M bar star, appearing in the expression for the offspring microbiome-mediated trait. And this is completely facilitated by environmental transmission, right? Thank you. And so, so we can have adaptation without any parent-offspring resemblance. I think that's a really huge result that we still obviously need to test empirically to see if this ever happens in nature. And so this is just one small, tiny little slice of this full project. If you want the rest of the details, check out our preprint. And I also have my email up here too if you want to contact me to talk about this stuff further. Thank you, everybody. This is the Eugene crew. Appreciate you all coming out here. Yes.
So is your vision for this that in, like, you're measuring the contribution of the entire microbiome, and if so, like, at what taxonomic level, like, by that, or is it, like, tracking a specific family of the microbiome, like, across its inheritance, or what kind of, like, taxonomic level do you foresee this being used for the microbes?
Yeah, that's a great question. I would say the whole taxonomic level. So we're interested in, like, it would be like which microbiome, right? So is it the gut microbiome? Is it the skin microbiome? Is it the whichever microbiome? So like focusing on that, and we'd ask like, how does that microbiome mediate some phenotype of the host, right? And so we would sample that microbiome, and then we would look at the composition of that microbiome across different host organisms and across host generations. Oh, well, I guess this is, yeah, I think it depends. Like, since I'm not an empiricist, I don't know. I know that there's issues with OTUs versus ASVs, for example, and I think they're saying that ASVs are becoming more in favor. So that sounds great to me. But yeah, I keep it very abstract and think about, like, just the composition of the microbiome and assume that somebody else has sorted out the taxonomic details. Yeah. Yes.
That's a great talk.
Thank you.
What percentage of phenotypic variation do you think is explained by the interaction of the microbiome?
I think it depends on the phenotype. In general, yeah, I don't know.
Large effects or the small effects?
Yeah, I don't know.
Genetics, for example.
Yeah, I think that's a really interesting question. I think that has a really big answer, right? Because it depends on when and where, which microbiome, which traits, and how do you measure it. The thing with the microbiomes is that they're so highly dimensional, is that they seem to explain everything, which is a little bit of a problem, right? So that's not necessarily a good answer for us. But then, yeah, thank you.
So good morning, everyone. Thank you so much for being here in my talk. So this is Maria Tocora from the University of Toronto. And my research, I'm going to talk about my research in the co-diversification of ants and microbes across the Galapagos Islands. So I hope you enjoyed the presentation, and I'm looking forward to answering your questions. So geographic barriers, like the ones that we see across the Galapagos archipelago, are conducive to population divergence while decreasing genetic flow across the space. So while studying interactions between ants and microbes, we might consider that in isolated systems, this might enhance this genetic differentiation as a strong local reciprocal oscillation reinforces the genotype by genotype interactions between partners. So after a long time, you might expect different lineages across the islands, as isolation has been a major determinant of this change. However, the transmission mode of these symbionts is also an important role in these interactions. As in vertically inherited microbes coming from parents to offspring, we might expect this interaction to be tighter as horizontal transmission is decreased. And in evolutionary time, we then expect having phylogenetic congruence, where the phylogeny of the microbes mirror the phylogeny of their host. So considering this, my research explores how microbes evolve in concert with their partners across space and time in the Galapagos Islands. So to explore these questions, I mainly focus on the ancient endosymbiosis between ants and their associated microbes. These ants are for the genus Camponorus, a really widespread genus with more than 1,000 species around the world. And these Leukemania protobacteria resides in specialized organs where they are suggested to be involved in different metabolic pathways. So in the Galapagos Islands, we have two endemic species of carpenter ants, Camponorus planus and Camponorus maculentus, with 11 and 13 subspecies described for the islands and differentially distributed across the archipelago. So this is really exciting since this is the first time that we are studying these species after 100 years ago that they were originally described. We are also including the species Camponorus sonarus, that is an introduced species to the Galapagos, also introduced in Bahamas and Jamaica, but native from South America. Including this species is important since we can explore different diversification-related mechanisms, considering the differences in the time scales over which these ants arrive into the Galapagos Islands. So first, for methods, we went to the Galapagos and started sampling these ants. That is actually quite hard since they really like residing in the roots of endangered and endemic cactus species in the islands. So sometimes you can find them hiding in the rocks or even foraging across different vegetation. Then we got the first de novo reference genome for a carpenter ant in the neotropics. So it's really exciting because it is coming from the Galapagos Islands. Then we did filtering and genotyping, calling variants for downstream analysis, such as population structure and also phylogenomics. As for the results, we're really proud to show this as the first de novo reference genome that is composed, as you can see in this high-seq contact heat map, for 26 scaffolds containing around 98% of the genomes. So this is a really high-quality genome for a Camponotus species. And also, we annotated this genome, mainly exploring the genome content by calling the coding sequences, as you can see in the first panel here, that they vary across the different scaffolds. We also explored TE and repetitive elements that actually are really high across the different scaffolds. This is surprising for Camponotus, and it has been actually previously described in other genera, such as Cardiocondyla and Solenopsis. And also, we explored this content across the scaffolds for this de novo assembly. The next step then was to explore the genomic data for the bacteria. So here you can see a taxa annotated Bob map plot that mainly shows the genome here, and also different reads coming from actually the bacteria. So since these are at endosymbiont, sequencing the genomes are actually easier than sequencing other microbes associated to insects. We were able to retrieve the genomes for the reference genomes for the 3 Blockmania strains associated to each of the species that we are studying. And as we see in other bacteria genomes, we have different peaks of GC and AT content. So then based on this genomic data and the variants that we call, we started exploring the population genomics. So here I'm gonna walk you through the different species, starting by Camponotus planus. So this is an endemic species, and here in the map you can find the results for the host. So we find mainly 3 clusters. The first one is coming from samples from the islands of Santa Cruz and San Cristobal that are closely located to each other. They are here. The second one is Isabella and Floreana that are actually located closely to each other. And the next one is from Santiago and Marchena that are located here. Marchena has never been connected to Santiago, so this is an interesting result. For the bacteria, we find similar results with different clusters corresponding to island proximity. So we find that they are clustering being on hooked together, they are closely located. For the following species, Camponotus maculentus, we found something different since we have the populations really apart from each other, with the exception for samples coming from the islands Marchena and Pinta that are clustered together. However, this is different for the endosymbiont associated to the ants, since we have the samples apart from each other. So more important and amazing information coming from the bacteria. For the introduced species, Camponotus sonatus, we found low genetic structure across their geographic range, since most of the samples cluster together. This is not actually that surprising considering that this is an introduced species, and while you compare taxa coming from the continent, they also cluster together with the taxa that is residing in the Galapagos Islands. However, here we find 2 samples that are far from this main group. And here, this might be suggesting different introduction events happening in the Galapagos. That is not just an important biological information, but also essential for future monitoring programs in the archipelago. We found similar results for data-associated endosymbionts, as we see here. So our next step was to explore the genetic differentiation using phylogenomics. So here is the phylogeny of the host viewed as a consensus phylogeny from more than 4,000 trees. So here we have the different species, starting with the species Camponotus sonatus, that is the introduced one. So we find a big polytomy here with all these samples clustering together. Again, when we compare this with continental samples, we find similar results. So this species is not actually that variable in their genetics. But with the endemic species, we find a ton of variation. So here we have Camponotus macelentus with different clades coming from the different islands and clustered together based on island proximity. Similar results with Camponotus planus. That actually, we find here 3 main clusters coming from Santa Cruz, San Cristobal, Floreana, Isabela, and Santiago and Marchena. It's important to consider that this pattern, like data behavior, might be coming from the way these ants have been historically dispersed. And interestingly, there is a ton of communication between these islands, not just that they were historically close together, but also human movement across these islands. So then we compare the phylogeny of the host and the phylogeny of the bacteria to explore for this phylogenetic congruence that is expected in these type of systems, considering that first, the bacteria is an endosymbiont and also is a vertically transmitted microbe. So here we compare the 2 phylogenies. As you can see, we found phylogenetic congruence happening, but it's important to notice that although we have similar branches, the length of these branches are different. So we have different evolutionary rates, especially if we consider that we are comparing an insect and bacteria. So their evolution might be different and driven by different mechanisms. So originally, my question was how microbes evolve in concern with their partners across space. So we found that in overall, the bacteria strain distances correlate with the genetic and geographic distances of their associated host. This is interesting and also require more and more genomic information to understand what is happening among islands, but also within islands. So that is my next step, that is actually going to the Galapagos and collect more populations, more sampling, and doing more resequencing to understand what is happening, especially in this system where we have different subspecies described for the Galapagos. So we don't actually not even know if we are talking about like different species. So here in this map, you can see the shade spots that in the Galapagos Islands, and they correspond to all the sites that we haven't sampled. So we still have a ton of work to do with the system. So I want to acknowledge everyone for being here and all my funding institutions, and especially the rangers associated to the Galapagos National Park for all the knowledge and support during my fieldwork. And also welcome everyone to stay in touch with me. This is a project that is just beginning. So we need more people and recruit more workers to work on this project. Thank you. So,
variables of genetic of geographic structure is used?
yeah, so for the geographic structure, what we mainly used was the host dispersion, like where are the hosts located. So since these are an endosymbionts located in the abdomen, so you can just know that they are going to be there in the islands with the host. That's what was for geographic. And for the genomics, what we used was variants. We call variants across their genomes, the host and the bacteria. And we then said, okay, we have these variants coming from that specific location. So that's why we did the correlation.
So geographic structure is presence, absence.
Yes. Yeah.
Do you have plans to look at ectosymbionts in your sequencing? Can you detect them?
Yes, I think that we can do it. We are doing long-read sequencing, so we can definitely do it. It might be a little bit difficult for the type of sampling that we have, so it's advised to have liquid nitrogen in the field to avoid contamination of the samples, but we cannot carry liquid nitrogen across the islands. So we are using RNA to do it, RNA later, so we might need to filter all those contaminants, but we can definitely do it for sure. Good. Finish.
I was interested in the phylogenetic congruence
Mhm.
analysis because that's typically for vertically transmitted symbionts often done with actually looking at the host mitochondria genome because they are co-transmitted. So while you've looked at the host nuclear genome, so I was interested in seeing if there are particular regions that seem to be kind of, that show like a particular compatibility with the symbiont, or that is.
Yes, so historically what people, the first markers used to show phylogenetic congruence were highly conserved markers for the bacteria and for the host. And they shown phylogenetic congruence because they were like highly conserved. But since we all have phylogenies at the genome level, we wanted to check like different markers and thousands of variants to check if we still have the same pattern of congruence across the phylogeny. I ran different tests with different markers, and we found the congruence, but then we scale up to having the genomic data, and we found the same congruence. So it's not just that just the mitochondrial genome is doing it, it's that also nuclear genes that are doing it. Thank you.
Okay, hi everyone, my name is Andy. I'm a postdoc at UC San Diego, and today I want to talk about microbial biogeography and comparing host-associated and free-living communities. So after going from islands, we're going to aquatic islands now, which is lakes. And yeah, I'll tell you about this. So give me 1 second, just one. Okay, good. So studying biogeography is important for many fields in the biology, like ecology, for evolution, but also for conservation biology, because it helps us understand the distribution of biodiversity and understand the mechanisms that generate and maintain biodiversity across the globe. There are many examples of like biogeographic patterns. For example, the latitudinal diversity gradient, where you have higher biodiversity at the equator, like lower towards the poles, or the elevational diversity gradient, where you generally have lower diversity at higher elevations. And another pattern is the distance decay of similarity, where communities that are geographically closer to each other are more similar. And like with increasing distance, they become more dissimilar. So we have a good understanding of those biogeographic patterns, but historically, many of those patterns have been observed in macroorganisms, like animals and plants mostly. So this raises the question of how universal those patterns are, and do they apply to microbes as well? So over the last 2 decades, I would say we have gained some understanding of microbial biogeography, and there's now a bunch of studies describing those similar patterns in microbes, both host-associated and free-living. But still, we have like way fewer studies compared to macroorganisms. And this is particularly true for host-associated communities, where we have very few studies. And then even for free-living bacteria, where we have like a better idea based on soil bacteria or bacterial plankton in water, still like the patterns are quite inconsistent. So there's still many open questions of like what shapes microbial biogeography, and especially like comparing host versus free communities, like in the same geographic context. So yes. The question of like why we should care about microbial biogeography, I guess I don't have to convince this room that microbes are important. We had like many examples this morning already about how microbes can affect different host phenotypes, host fitness, but they also have like ecosystem-wide effects. So like in ecosystems, free-living bacteria can like regulate decomposition, nitrogen cycling have like many important effects. And to understand those effects for the ecosystem, but also for the host, we should also understand what drives their geographic distribution. Okay, and I want to introduce some questions that I'm trying to answer with this work is, first of all, do biogeographic patterns differ between host and free living communities? And another question is, what are the factors that affect microbial biogeography, and do different factors affect free living versus host-associated communities differently? And some factors might be more neutral factors like dispersal or ecological drift, which is more random or neutral, like I said, or the other like non-neutral processes like the host or the environment filtering or selecting for certain bacterial lineages. And the pattern I'm going to talk about today is the distance decay of similarity, and I'm going to contrast free living versus host-associated communities. And a few words about our study systems. First, we had free spine sticklebacks from 20 lakes on Vancouver Island in Canada. And for those, we sampled their guts and sequenced the microbiota based on 16S. And also disclaimer, I'm going to talk about bacterial communities only for this talk. And the geographic distance range from 0.1 kilometers to up to 80 kilometers. There's quite a range of geographic distance between different lakes that allows us to study this distance decay pattern. The other system is brook trout from the Sierra Nevada mountains in California. For this, we have 15 lakes. We sampled feces for those, and the geographic distance is, the range is not as big, but also from 0.1 to 35 kilometers, so quite some geographic variation. All right, and by contrasting those two systems, I hope it will help us understand if there's some general patterns that consistent across those two systems, and help us to infer how general those potential differences between host-associated and free-living communities are. I'm going to test, or we tested 3 hypotheses, starting with the first. So before we go into this biogeographic analysis, we need to establish whether those communities really differ, like host-associated versus free-living. And we hypothesized that there's a clustering by sample type, that they do actually differ, and they're not clustered by geography. So the water samples of one region are not the most similar to the fish of this same region, but rather the fish are more similar to each other than the water samples. So there's some host filtering by those fish, and also I guess the niche availability differs quite a bit between a host gut and a lake. Okay. So, and here are the results. So this is a PCOA plot, and you'll see the 2 different systems in colors and the sample type in the shapes, and I think it's really hard to see, so I summarized this for you. So over there on the left, you can see all the water samples from the Sierra Nevada and Vancouver Island, and on the right, you have all the fish samples, and when you do some statistics, you find that the sample type is the actual main determinant of microbiome composition. So there is clustering by sample type. All right, and to further show this, we also looked at the relative abundance of different bacterial phyla. So these are the main bacterial phyla in our data set. And we looked at differential abundance between host-associated and free-living communities across those 2 systems. And so the more values on the left, the more abundant this bacterial phyla is in fish guts. The more on the right, the more abundant it is in lake water. And one thing you can see is there's quite some consistency between the 2 systems. So for example, the Firmicutes on top, they are more abundant in fish guts in both systems. You can see those data points close together, and on the other end of the spectrum, there's the Bacteroidota, sorry, which are more abundant in the water. Those patterns are quite consistent. So we see that host-associated and free-living communities show this parallel divergence, and this might not be unexpected because we know that the host bacteria are not merely like a subset of what is found in the water or the environment. But I think it's still important to establish this, that we then can compare those biogeographic patterns. So yes, so the first hypothesis was confirmed. Now we looked at within lake community similarity. So how similar are samples from the same lake to each other, like how 2 fish from the same lake, how similar are they to each other, or 2 water samples from the same lake. And we hypothesized that water samples would be more homogenized. So across a lake, samples would be very similar, but the fish would be more different from each other compared to the water, because there might be more dispersal limitation between those different hosts, but also there might be host filtering. Also, disclaimer, I'm not differentiating between the neutral and non-neutral components in this part of the talk. So we can't right now. Okay, here are the results. So you'll see the community similarity on the y-axis, and we looked at lake water versus fish guts. And remember, this is how similar fish or water samples of the same lake are across all the different lakes. And what we find is that the communities of the fish guts are more different from each other compared to the water samples. So that's what we expected. More homogenized water samples, fish are more different from each other. And we find the same pattern, even much more pronounced in the stickleback system from Vancouver Island. Yeah, so similar patterns across those 2 systems. So this really shows that there's more divergence among those fish, meaning those fish represent islands within those lakes, and that the free-living communities are more homogeneous. And so at this point, we established that those communities are different from each other. Also, the within-lake community dynamics are different. And now we're finally looking at the biogeographic pattern across lakes. For this, we have two hypotheses. One would be that the gut microbiota would show like a stronger distance decay pattern because of dispersal limitation. So you could imagine that like free-living bacteria that live in the water might be more easily dispersed across those lakes, where it might be harder for a gut bacteria that's within a host to disperse between those lakes that are not connected. So these are distinct lakes and not connected at all by any rivers. On the other hand, we could hypothesize that the host might buffer the microbial community from environmental variation. So maybe if the host selects for a certain subset of bacteria, and those hosts are like all of the same species, they're closely related, so you might expect that there's not variation associated with geography because the host kind of maintains a similar microbiome across those lakes, which would be like more host filtering or non-neutral processes. Okay, let's get into the results. Starting with the Sierra Nevada brook trout system first. Again, you will see community similarity on the y-axis, and now on the x-axis there's a geographic distance. So what we find is that there's stronger distance decay in the free-living communities on the left. You can see that the slope is steeper. We also did Mantel tests, and we found that there's a stronger correlation between community similarity and geographic distance. So, and you also probably see that there's a lot of variation, especially in the gut microbiomes, like even in very geographically close lakes, there's a lot of variation. So fish from two very close lakes could be like very dissimilar, whereas like fish from like far apart lakes could be quite similar. But overall, the trends are weaker. And then we find the same pattern for stickleback fish. Again, higher distance decay in the free-living communities and less of an effect in the stickleback. All right, so for the third hypothesis, we found evidence that distance decay rates are weaker in the gut microbiota. So that means that there's probably like those non-neutral processes of host filtering have a stronger effect. So yeah, which again means that those communities are shielded from the environment and to a certain extent, whereas like the free living communities are more sensitive to spatial and environmental gradients. Okay, to come to the conclusions, we found that free living and host-associated communities are differentially affected by neutral and non-neutral processes, which leads to variation in their biogeography. All right, and then, I mean, this is one study. There's like we had some studies looking at biogeographic patterns already today, and I think there's a lot of work to be done. Some questions that could be addressed in the future and some considerations are like, what about other biogeographic patterns, you know, latitudinal gradients, elevational gradients, I mentioned those already, or diversity area relationship. Do we find like differential patterns between free living versus host associated communities across those different patterns? Also, what about microbiomes with like different exposure to external environments? Like if you think about the gill versus gut, the exposure to the environment is quite different. And also, what about spatial scales? You know, like at smaller, bigger spatial scales can also inform us about those patterns. And yes, and I think ultimately coming back to what this actually means and why we should care, I think understanding this geographic variation, those microbial communities, okay, thanks, could affect ecosystem ecology, but also like could affect the host ecology and evolution. Again, we heard those examples already today, how microbes are important for their hosts. I think if we will fully understand how they affect host ecology and evolution, we also have to understand what shapes the biogeographic distribution of those bacterial communities. And with this, I would like to finish this talk. I want to thank my co-authors, my funding agencies, and specifically I want to highlight Josh Dominguez. He is leading this project. He's a very talented grad student at UCSD. And I also want to thank Diana Rennison, my advisor, for her support. And yeah, I want to thank you for listening. Thanks. Yeah.
So I have a question on those distance decay relationships. So you had the hypothesis that there was like a host filtering would reduce the distance decay relationships, but then the community similarity index is like extremely variable and often very, very, very low.
Mm-hmm.
But wouldn't we expect the community similarity to be high under that hypothesis? So is there an alternative hypothesis that might match better with the data?
I agree with you, and I've been thinking about this a lot, and I think it's confusing because we do see much more variation within each lake and like across like close lakes. You know, you saw this in this plot, right? But it's not structured geographically. So like even like within the same lake, which I didn't show this, so this distance decay did not show within lake comparisons, but like even within the same lake, you see like this spread from like basically being completely different to being completely similar. So there is a lot of variation within and across lakes, but it's not an effective geographic distance. So I would argue that they are shielded from this environmental gradients that might exist, but I don't understand fully like why there's so much variation within each lake. So what shapes, like, you know, is it diet? Is it just random? You know, is it like priority effects? So we haven't measured this. So I think this is still a mystery to me of like why there's so much variation. Also, sorry, as a side note, people study core microbiomes, and like in different species, you find like different strength of core microbiomes. We don't find this at all in the stickleback, for example. Like they're so variable, and yeah, I don't fully understand. We can talk about it more. Yeah.
Hi, I'm Andy. I'm from Exeter. They keep all their evolutionary ecologists down in Cornwall. I'm going to bang on about snails. This is a talk about the common garden snail and its microbiota, but I think it's worth at least acknowledging that the marine Symbioses people definitely have all the big, well-trammeled model charismatic organisms. Bobtail squid, these amazing sea hare things that kleptoplasty and redeploy the stinging cells from sea anemones and put them in these amazing serrata. They're kind of really well understood in a natural history sense, and the same is not necessarily true of terrestrial mollusks. And this, so the takeaway message from this slide is that this is a 100-year-old hypothesis that kind of came out of a physics innovation and was left to us by this French guy. And there's a huge amount of interesting research to talk about in the middle here, but maybe people could ask me questions about it. But the thing was that this as a hypothesis was really nailed in the 1970s in Chicago. But people publish in esoteric malacology journals, and no one reads them. This is what that French guy kind of would have seen when he brought a snail. This is actually a snail I rescued out of a neighbour's beer trap in his back garden. It's not irritated, it's just drunk. You get this bright canary yellow mucus, which under blue light is luridly fluorescent green. And because none of my friends really asked me, Andy, what happens when you poke a snail, I made a video. So this is slowed down to kind of a quarter speed. That's an entomology pin and my clumsy finger, and we're poking this snail in the mantle. So it's trying to have a snooze, it's estivating. And you can see there's these spreading zones of bright fluorescence that kind of play out over the mantle. As a behavior, I've slowed this down because it all happens quite quickly. And it will eventually pivot round to its side view, and you'll see this very devil's own cauldron of foamy fondue fluorescence. And what the snail is now doing is it's pumping bubbles into that mucus layer through its pneumatostome. And this is referred to as being it going into alarm. Not all snails do this, and in some respects, snails kind of really rely on mucus because it's what keeps their kind of wet mucosa from being entirely confluent with their environment. And there's also a whole ton of things that want to eat them. So this is, in some respects, this snail saying, "Oh, get off me." Hopefully it will end. Come on now. I think I'll end it. So we've had a lot of fun in the last couple of years looking more closely at that mucus. And so at least some of what we've done is kind of flow cytometry characterizing it. And it is incredibly dense with bacteria. So there's kind of nearly 200 or so thousand bacterial cells per microliter in that mucus. There are around 100,000 of these host immune cells that stain with nuclease 468. And then there are far fewer again of these much larger bacteriocyst cells. Roughly 10% of those cells will be fluorescent. We kind of characterize that on two systems, the Attune NXT site picks and the image stream. So 10% of those cells are fluorescent. And when you look at the mucus under the microscope, you also get this feeling that there are bacteria which are and are not fluorescent. And when you come to genotype them, so these are all isolates, and fully half of this phylogeny is all Pseudomonas, but there's really strong evidence of geographic substructuring. And even within snails, there's very high variance in traits. So these are snails with parviflua, Pseudomonas parviflua, so they're from a St. Andrews collected snail. And they have a very large 3 order magnitude type variance in their siderophore production. I'm now one of these people that collects snails wherever they go on holiday. On the right here, there's snails from Versailles, and then some in the middle from Penryn in Cornwall, some from the Harbour Wall in Leverborough, and some from Wales and various other little places. And this is 16S nanopore whole 16S sequencing. So we can get much better resolution now on what isolates are actually, what communities rather, are what in what strains. And a kind of, as a rough breakdown, we have at the genus level Pseudomonas written through the core of the microbiome. And I'll move on to talk about what we've done in terms of trying to understand the transmission of the microbes and whether or not they can be adequately displaced and what they might be contributing to the host in terms of fitness. Neonate snails are tiny. You can put them in homogenization tubes, and you can plate out and count the bacteria, so standard dilution plating. Snails are not stupid. If you try and clear them of their infection by feeding them high doses of antibiotic, they really don't like it. So the rifampicin dose down at the bottom there, that bright orange media, this is a standard autoclaved snail porridge sort of thing. If you lace it with antibiotics, they do quite strong diet avoidance. And that's because at their intermediate doses, where they're not doing the diet avoidance and getting selection against their microbiomes, whilst you see plenty of clearance, you also end up with lots of death. Baby snails are really cute, so we didn't do very many of these experiments. It's a bit grueling, killing lots of snails. But we did try and reinvade with rifampicin-resistant spontaneous mutants. And you can see here that we kind of recover some survivorship and protection, or at least strain 2 seems to in the top right of this panel, as does the strain mix, which is just basically all of them put back in. But for the most part, snails are unhappy and die a lot when you try and get rid of the microbiome. And this is at least consistent and speaks to what maybe is the core function of this microbiome, which is it's sort of sat there in the cuticle in these big club-shaped glandular cells as a defensive microbiome, effectively a microbial-mediated immune system. So this on the left here is what these tissues look like in section, and the bright fluorescent dots I would put to you are Pseudomonas living in these big cells. They're about 200 micron long. When you, on the right here, feed a snail lots of gentamicin and a gentamicin-resistant red fluorescent protein-expressing strain, you get this partial regression of the resident infection and some kind of moderate invasion by the red strain, but you do not get displacement. And this is kind of consistent again with it being kind of an immune system. But snails do loads of other stuff, which mucus is really important for. So this is a picture of a snail taken through a glass slide, and you can see this very bright fluorescent ring around the outside here. That's the epiphragm. It's simultaneously like a glue and a do not disturb sign. And snails use mucus a lot to kind of maneuver their way around their environment. But this alarm response, we think, is at least a response to defense, probably against beetles and other determined predators. Snails turn out to be delicious. They don't really decompose, they just liquefy into delicious gloop. So these carabid beetles are always out trying to eat them. What's true of the alarm response is that it is not well tolerated by snails. So if you get students to poke snails, twice a week for a month, their mean survival is 0. They all die. And you can see that the survivorship tails off precipitously the more you poke them. But this is also true that as a behavior seems to take quite a long time to recharge. So snail's mucal volume, again, drops off a bit of a cliff. So the first alarm response is quite a high volume one, and all subsequent responses are quite low in relative volume. And you can do this experiment the other way around. So you all know the Queen theme for Flash, with that kind of backbeat. Actually, it's faster than that. It's more like Flash. So if you poke a snail at about 2 beats a second, all of the snails, all 50 or so of them, will go into alarm within the first 20 seconds. If you slow that right down, quite a lot of the snails will really ride it out and also then not go into alarm. So this behavior is on kind of a trigger. Because it is so costly, the snails are exercising quite a lot of restraint over when they do and do not go into alarm. So this is a 100-year-old hypothesis with some Sleeping Beauty papers 50 years later. It's sort of largely ignored, but how general these sorts of things are with mollusks, because all of the interesting marine ones are all cuticle-born, is an open question. And I think there's 200 years worth of natural history here on the autoecology of the snail, of snails generally, but they're kind of ignoring perhaps the microbiome in the room. And I've got provocative colleagues that always say to me, Andrew, who cares? Everything's got a microbiome. I think that kind of as an attitude comes at the cost of perhaps misunderstanding quite a lot of the other ecology. So a lot of this work is just not me at all, but project students. And at least one of these people, it's false advertising. They're not actually an extra student. So Arya Nagarajan there is a really talented natural history photography student from Falmouth Uni doing a kind of an arts degree. And she's responsible for that video and some of the nicer photos. Rachel, looking a bit craven here, is the chief snail poker. And the rest of them have done sterling work with very short internships. And everyone else just puts up with me. Okay. Yeah, the trail mucus is not as fluorescent at all. It really is their alarm mucus that is garishly lurid. Suggesting that there is different kinds of mucus. So there's a whole field, mucomics, it's a disgusting word, but there's a whole field that looks at different kinds of mucus from snails, and this is a model organism for that. But the degree to which these people have picked up on the fact that the mucus en masse is really a big, dense microbial biofilm is probably a bit limited. The fluorescence in question is siderophore. It's probably the dissociated phenazine fluorophore. So there's the degree to which any of that is adaptive is a bit up for grabs as well. I suspect not. I think it's just like when you ask the Pseudomonas people why is siderophore fluorescent, they've got no idea. I don't think it's anything. I think that's all by the by. So we've got data that I didn't have time to show, but which you might have a slide for. Come on. Right. There's way too much data on this slide. But the takeaway message is that if you feed syringe-filtered mucus to other bacteria across the temperature gradient, you can get quite strong strain-specific inhibition. So you can definitely dial down other microbes' growth via the secretions, independent of the bacteria being in them. but it probably, I think the main effect is a priority effect. So you're talking about immune priming, but it's more about having a resident and it being hard to dislodge.
Hi, everyone. I'm Svitlana Serga, and I'm a researcher from Ukraine, actually working in France. And it's my pleasure to present our work, which has long history, and it was started in Ukraine and have great development in the lab in France. I would tell about biological invasions. Biological invasions are processes of invasion of species to a new environment and establish new populations. And in this case, we have native populations. We have native populations and invasive populations. And now we have great information about evolutionary dynamics of host under invasion process. And usually we have reduction of host genetic diversity in invasive populations. And then some alleles could be fixed or lost because of selection or because of genetic drift. But what we know about symbiotic bacteria in such processes, especially vertically transmitted symbionts. And we, in theory, we expect the same situation, that symbiotic bacteria could be fixed in population or could be lost. And one of the great examples of vertically transmitted symbionts are reproductive parasites, and they are even more interesting in such topic because they can bias their transmission and biased host populations because they transmitted only maternally or predominantly maternally, and it means that all progeny in infected females are infected too. And to spread in the populations, such bacteria could develop mechanisms to increase number of infected females in the populations. And one way to do this was described as cytoplasmic incompatibility. It means that in crosses of uninfected females with infected males, we can see embryonic lethality. It means that there is no progeny. But in progeny of infected females, we can see normal development as well as in progeny of uninfected females and males. And such phenotype is very often in insect populations, especially in Drosophila taxa, and great example of such phenotype is in Drosophila suzuki, spotted winged Drosophila, which is invasive species and also pest species. Drosophila suzuki is originated from Southeast Asia, namely from China, and we have recent invasion, which started in 2008, and we have two different routes of invasion, one in America and the other one in Europe, and then it spread quickly to South America and Africa. And we tested as measure of evolutionary dynamics of symbiont, we measure Wolbachia prevalence in invaded and native areas. And in native areas, we found high infection frequency of the bacteria. But in invasive areas, we found decrease in infection frequency. And such decrease is different in two differently invaded areas, in Europe and in America. In America, we have a great decrease in infection frequencies. So we saw a great reduction in prevalence in invasive populations. And the main question, why we have such situation? And Wolbachia population dynamics can be described using three parameters. It's a rate of maternal transmission, which I described already. the reduction in relative hatch rate, this parameter can display cytoplasmic incompatibility and the reduction of fitness of infected females. We can expect the same situation in invaded area as in the host. Infection frequency could be fixed or could be lost. But if we have cytoplasmic incompatibility, we have a threshold which we called p hat. And p hat depends on two parameters. If maternal transmission is perfect, it's dependent on reduction of relative fitness or reduction in hatch rate. So if we have great reduction of hatch rate, we have highest threshold. Lowest threshold, sorry. And if we have a great reduction in fitness, we have highest threshold. And we wanted to test how we can describe such parameter in populations of Drosophila suzuki. To do this, we used laboratory strains of Drosophila suzuki as more as we could find in different laboratories. And we tested 53 strains of Drosophila suzuki and found 15 strains infected with Wolbachia. And then we performed cytoplasmic incompatibility assay in such strains. We made all 4 possible crosses for each strain between infected and uninfected strains. We made this for strains from different areas, from native and invasive. And what we found, you can see in the graph hatch rate in y-axis and different types of crosses corresponding to the graph. And the main interesting is a test cross or incompatible cross when we cross uninfected female with infected male. And if we see a reduction in hatch rate, it means the strain could induce CI, cytoplasmic incompatibility. And we found such a strain in European populations, and in one strain we found even more reduction in hatch rate. But we found this not in all strains from Europe. So we have great variation in the level of cytoplasmic incompatibility in European populations. In American populations, we found reduction in test cross in 2 strains, but also we found a reduction in hatch rate in control cross. It means that we have male genetic effect, but we need to test this more closely. And the same situation we found in Asian populations from China, but we didn't find a high level of cytoplasmic incompatibility in other strains from native range from China. So we have no difference in cytoplasmic incompatibility between native and invasive population, which we expected. And also in the same types of crosses, we measured fecundity as number of eggs laid by females. And to simplify this experiment, I used only I show only progeny from a cross of infected female and uninfected female with uninfected male. And we really didn't find a great reduction in fecundity in infected individuals. So we can conclude that there is no deleterious Wolbachia effects in the laboratory. But I need to mention that we performed all experiments in the laboratory. In nature, it could be a slightly different picture. And so we can conclude that there is no difference in p hat in native and invasive populations, which we firstly expected. And we can describe difference in infection frequency by possibly by drift, and native populations are under this threshold, and invasive populations because of drift are below such threshold. And in conclusion, we found low infection frequencies in invasive populations and in native, and we found no difference in cytoplasmic incompatibility between native and invasive populations, and no deleterious effects on fecundity. In perspective, we need to measure transmission fidelity to confirm that such effect is not due to differences in transmission fidelity, maternal transmission fidelity. And we want to estimate both parameters in nature because it could be quite different from the lab. But technically it's difficult to do because native populations are in China, and we need to do this in China. And I want to say, acknowledge all people who are involved in this work. It was started in Ukraine under supervision of Irina Kozeretska, and also in Finland with Emily Knott. And now I'm working at CBGP in INRA with Nicolas Roth, and he supervised mostly such work. And also I want to say thank you, Matthew Gauthier and Arno Stup, and students working on such project, and also technicians from the lab, and people who gave flies, because it was great work to collect such flies. And also thank you to my funding, PAUS program, and Maria Skladovska-Curie for Ukraine. And thank you for your attention. Yeah, it's a great question. Thank you. We performed sequencing, Illumina sequencing of Drosophila Suzuki from different locations, and we found actually 3 clusters of Wolbachia corresponding to the invasion routes. Like we have Asian type of Wolbachia, we have American type of Wolbachia, and we have European type of Wolbachia. But the most interesting thing is that we never have American type of Wolbachia in Europe and European type of Wolbachia in America, despite some admixture events between 2 invasion routes. Yes?
So these invasive places are environmentally very different from each other. Did you look at any sort of environmental variables, temperature kind of factors that might affect the Wolbachia?
Yeah, thank you for this question. We started to do such analysis. We tested 9 environmental variables, which were mean of months before collection in each site where we can have such information, and we didn't find any correlation with environmental variables. So I think we have more influence of other factors on the infection rate.
Really, really nice talk.
Thank you.
Have you looked for, or do you plan to look for, like other sort of endosymbionts of Wolbachia that have cytoplasmic group and spiroplasma or anything like that?
Yeah, thank you for the question. In Drosophila, other symbiont is very frequent, spiroplasma. Its symbiont can induce not cytoplasmic incompatibility, but male killing. And we checked our strains which we tested, and also the data from natural populations for other symbionts, and we didn't find any other symbionts in the data.
Related to the first question, that you described Wolbachia has 3 different types, and how about the Drosophila host type? Have you considered genetic distance of the host, the combination of the genotype, Wolbachia genotype, predicting the sequence?
Yes, thank you for the question. We made such analysis using mitochondrial DNA data, and we have the same pattern for mitochondrial data, and also we have similar clusters for whole genome data too.
I was just curious, do you think the lower infection rate in the invasive populations actually helps them to be invasive, to invade other areas?
Sorry, I didn't understand actually what...
The invasive populations have a lower infection frequency, and
Yes.
I was wondering if that's actually beneficial for them to basically be able to be an invasive species?
It's a great question, but it's really difficult to measure fitness benefits, especially in nature. So we need to find a good way to compare fitness benefits of infected and uninfected individuals in different areas. And we want to do this next, because we started from the lab, it's easier, and then we want to check this. Yeah, thank you.
Hello, my name is Laura Lardinois. I'm a PhD student here at McGill in Montreal, although most recently I've spent most of my time in Panama, where I work at the Smithsonian Tropical Research Institute, and that's where I do my fieldwork, and this is where the data you'll see is coming from today. So first, I'd like you to meet two of my study species, Abudafduf saxatilis and Abudafduf tischelii. These are a pair of damselfishes, and while they look really, really similar, they actually live in two different oceans. So saxatilis is found in coral reefs of the Caribbean, while tischelii is found in the Pacific. But there is a reason why they look so similar, and this is that they're the great-great-great-great-grandchildren of a single population that was split by the formation of the Isthmus of Panama around 3 million years ago. And we know these as sister species or geminate species, and they're an amazing model system to look at diversion evolution as well as adaptation to changing environments. And in my case, I'm interested in not only the fish's adaptation, but also that of their microbiomes. So in terms of differences in environments, in the Caribbean, the temperatures are warmer, more saline, and nutrient poor, while in the Pacific, you have more variable, cooler temperatures. There's upwelling, especially in turquoise, you see the Gulf of Panama, where there's seasonal upwelling that brings major temperature shifts. It goes from around 29 degrees Celsius, which is the usual, down to 16 degrees, and that can happen in a matter of hours, and it fluctuates quite a bit, and this happens every single year. This also brings rich nutrients, so the Pacific has a much higher primary productivity, and it's less saline. The other thing is these environmental changes also mean that the reefscape is very different. So in the Caribbean, you have high coral diversity and complexity, while in the Pacific, you have a dominance of these more stress-resistant cauliflower corals or Pacillopora. And I'm interested in what the sister species can help us learn about the ecological and evolutionary factors that drive microbiome composition. And by comparing these sister species, we can look at whether factors linked to the host and host phylogeny and genetic relationships, or more broad environmental characteristics are the stronger drivers for these microbial communities. So we'd expect that if the environment has a bigger role, you'd see these big clusters by ocean. So you'd have a Pacific group and a Caribbean group, especially considering that it's been millions of years since these populations have been separated. And you might see small clusters by host species, but in general, like a big separation by ocean. On the other hand, if there's a stronger influence of host, both in terms of host filtering and selection, we might actually see clusters by sister species, so that these pairs of fish species or other hosts would still have relatively conserved microbiomes. And to test this, we looked at 7 different sister species pairs. So these cover a whole range of trophic groups, because diet can also have a large influence on the microbiome. So from left to right, you've got herbivores through carnivores. We collected them in the 2 different oceans and from 2 different sites, so trying to look at variability at that level. We collected both skin swabs in the field as well as performing dissections to get hindgut mucus samples for each fish, and did 16S rRNA sequencing to look at the microbial communities. So here are some of the preliminary data. This is gut microbiome, and we can see that already it doesn't cluster by ocean, but rather by sister species pair. There's also clearly an influence of diet. So in green, you've got the herbivores. Up there, you've got more of the omnivores and carnivores, and then all the ones that are more like invertivores and spongivores kind of go to the left side of the screen. What I found was super interesting with these is also that like certain pairs clustered much more tightly than others. And I think to some extent, that separation that you see with more of herbivores and invertivores might be that these are more complex things to digest, and so they have a more tightly linked relationship with their microbiome. So yeah, in some with here, we're seeing that these sister fish gut microbiomes are clearly clustering together, and that trophic group also matters with kind of more of an overlap with omnivores and carnivores and more of a separation with some of these other trophic groups. So this is more supporting this idea that the evolutionary relationships and the host is a stronger driver of this gut microbiome community. And when we look more at the microbial phyla composing it, again, each vertical bar is a host species, and they're paired by a sister pair. So if we focus, for instance, on these are a pair of angelfishes, and we can see that they have this much larger proportion of Bacteroideota, which to me was really interesting because this is known as a symbiont for a lot of other hosts in the gut microbiome. So these are also really interesting fish in that they are almost exclusively spongivores. So they had a very different diet, and also you can smell when you open their guts that they're clearly relying on some sort of microbial fermentation to digest these things. So looking at the phyla in these sister fish, again, we're seeing kind of this variation between hosts, but that they're most similar between sister pairs rather than having kind of more of a conserved base microbiome by ocean. And again, some potential key symbionts that are helping with digestion at this level. So in some, we found that our sister fish had very similar microbial communities despite this separation, and that it was not driven strongly by the different reef environments, which indicates to me that host species and these evolutionary relationships are probably outweighing the external environment in structuring at least the fish gut microbiome, suggesting strong host microbe relationships that are conserved despite major environmental changes. And again, like I'm saying, these are pretty preliminary results. So in terms of next steps, I really want to test for co-phylogeny and co-diversification and seeing exactly to what level these relationships are conserved, and also what sorts of functional roles these microbes might be playing for their hosts. And with that, I'll take any questions.
That was a very interesting talk. So is there any mechanism for passing down the microbiome from generation?
Yeah, so that's the super cool thing is we know for most of these fish, the eggs are aposymbiotic, so they don't actually have direct vertical transmission of microbes from the parents. To some extent, though, I do think that parents could transfer, especially in like damselfishes where you see them kind of guarding nests and stuff like that. So like in the initial stages of life, there is kind of that contact between like others of that same species and the eggs that will then eventually develop. But a lot of it has to come from the environment because you don't have that same level of like thing you would have in a mammal. So to me, that's also super interesting that we have these very different environments that are still managing to kind of like acquire and select for a very host-specific microbial community. That's a good point. Yeah, so in this PCA in particular, I narrowed it down to only the true sister species pairs. And you're right that, like, in this case, for instance, the herbivores are both within the same family, but I have a bigger pool of samples that include other herbivores that aren't at all closely related, and they still cluster more. So I haven't explicitly controlled for phylogeny, but you still see these kind of patterns where even very distantly related fish will cluster more by diet. And the other piece of this project that one of my collaborators is working on is actually doing a lot of diet metabarcoding to get more fine-tuned data on all of these individual fish. So we'll be able to actually physically test for diet, phylogeny, and microbiome together. Thank you. I saw a hand there.
I'm wondering if within sister species pairs, you've done any differential abundance testing of the microbiomes of that pair?
Like comparing, yeah. I have not. That's definitely on my to-do list is, kind of picking apart within these pairs, especially because from a first glance, it looks like some of them are much closer than others in terms of big picture microbial community, and kind of that would then let us get at a finer scale, kind of seeing what's going on there. Yeah, thank you.
So you mentioned that you had skin samples. Have you analyzed those? And I wonder if there's a bigger environmental effect there.
Yeah, so I'm still in the process of analyzing those. But I do have skin samples from, I've basically analyzed half of it, which is why I didn't show it yet. The environment definitely has a stronger influence of it, but I was surprised by how much the host still has an impact. Because these are surface microbiomes. They're in constant contact with the water. And we do also have water samples to compare with this. And it's very unique compared to the water. So it's definitely shaped by the host and by kind of this relationship. And again, the mucus itself is not necessarily, on a microbial level, is a whole nother environment in and of itself.
So this is kind of following up on that question and are any of these, do any of these fish do the mouth brooding of these eggs?
No.
Well, that would be interesting in those species to look at oral microbiomes.
Yeah, no, definitely. None of these particular species do, and a lot of these species have the very, very bare minimum of they lay eggs and they leave. Some of them stick around a bit more.
I'm curious if you found differences between Bocas and the other Caribbean side, and between Coiba and the Gulf of Mexico and the Gulf of Panama.
Yeah, so that's another thing that I need to dig into more is big picture, there's clear differences between host species and things like that. But between our sites, we also see between some of the sites it's significant differences, some of the sites it's not. And I need to check exactly why that is. I'm not surprised. There's a significant difference between Bocas and Portobello, where we collected, which for those who haven't been to those sites, that does not surprise me at all, because the reef communities, especially in Bocas where we were sampling, have been really heavily impacted, and the fish there were, I think, highly stressed and much smaller actually than what we were seeing in Portobello. So there's definitely human impact as well that's having an impact on these communities.